Hi, I'm Natalie Castellana, CEO at Abterra Bio. For almost 20 years, I've been developing tools to analyze antibody proteins directly, and I'm excited to share with you our newest technology, Griffin, for determining polyclonal antibody sequences and unlocking the serum antibody repertoire. If you work in antibody discovery, then you're likely familiar with technologies like hybridomas, single B-cell screening, and phage display to access the antibody repertoire encoded in B cells. But B cells are only part of the story. Antibodies secreted in serum are the active molecules fighting pathogens, causing autoimmune disorders, or giving our titer readout. B-cell-focused approaches to antibody discovery use the serum titer readout for decision-making, but ultimately the serum is thrown away. In this talk, I'll convince you that serum is an ideal source for novel antibodies. There's very good reason to focus your antibody discovery campaigns on serum. There's 11 orders of magnitude more antibodies in blood than B cells. That's the difference between grains of sand in a bucket and grains of sand in all the beaches in the world. And I should know, we're based in San Diego. Even if you account for a difference in mass, there's still 8 orders of magnitude more antibody mass in blood than there are B cells. That density of antibodies in blood means that we can screen an extremely large number, millions of antibodies, from what is essentially a thimble of serum. From our years of experience analyzing the immune response, even when the B-cell repertoire is accessible, we find that it's an imperfect proxy for the antibodies in serum. Only about 1% of antigen-sorted B cells in blood are contributing to the serum antibody repertoire. If you are only looking at B cells, you're also missing out on at least a third of the antibodies in serum. In our experience, at least 30% of the clones in serum are not encoded in a captured B cell in the repertoire. For many of the projects that we work on with Griffin, a B cell-focused approach is not possible at all. If you're working with retrospective samples, for example, you're often volume limited, cell limited, and viability limited. If you're working in non-standard host species, there's a dearth of tools for targeting B cells. If you're working in patients with latent disease who have long since recovered from the acute phase of the illness, then B cells are going to be very rare. If you're looking at non-blood biofluids like cerebral spinal fluid, the B cells will be basically non-existent. And of course, for most polyclonals that are used in kits and in diagnostics, there are no B cells available. Griffin is currently being used for a variety of applications, from monoclonal antibody sequencing discovery from animals as well as directly from patients, for vaccine research, for tracking clones across time for monitoring minimal residual disease, and for replacing polyclonal antibodies in diagnostics and kits with recombinant mixes that are well characterized and reproducible. So how does it work? Well, we start with plasma or serum to generate an antigen-purified polyclonal antibody using affinity chromatography. We then subject that polyclonal antibody to no less than 10 enzymatic digests and LC-MS/MS runs. We divide the Griffin process into 3 stages. In the first stage, we assess the complexity of the sample by cataloging the clones and doing a clonality assessment. This stage helps us de-risk the project and determine if simplification is needed prior to sequencing. We use our own proprietary de novo peptide sequencing tool to determine the peptide sequences directly from the mass spectrometry data without a reference database. We extract the CDR3 sequences from that peptide set and we construct a CDR3 network. Each node in this network is a unique CDR3 sequence, and the edges are drawn between CDR3s that are within an edit distance tolerance. We can apply the clonality assessment to a variety of samples with ranging complexity. On one end, we have monoclonal antibodies, which only contain a small or hopefully one CDR3 sequence. On the other end is a natural human immune response to a pathogen where the antibodies are targeting many epitopes. In between are moderate complexity samples like mixes of monoclonal antibodies or a hyperimmunized animal. And Griffin can be used to sequence all of these cases. After clonality assessment, in stage 2, we generate full-length antibody sequences for each identified CDR3 from the previous step. And in the final stage, we express and test a panel of candidate monoclonal antibodies to recapitulate the activity of the starting polyclonal. To illustrate how this works in practice, let's go through a case study that we just completed. Dr. Thomas Kukar's lab at Emory University had developed a rabbit polyclonal antibody to several different domains of progranulin, a pleiotropic protein implicated in neurodegenerative diseases. The supply of polyclonal was running low, and immunizing new rabbits is a lengthy process and risky, since the resulting polyclonal may have very different properties from the original. Since no B cells were available, we collaborated with Dr. Kukar to use Griffin to create recombinant antibodies to the granulin 2 domain. After purifying the rabbit serum against the granulin 2 domain, we generated mass spectrometry data and performed the stage 1 clonality assessment. This is what the network looks like. A network component with multiple CDR3s is often a strong signal that the CDR3 is abundant in the polyclonal and was potentially undergoing affinity maturation, resulting in several variant clones. Individual CDR3s can be assessed for quality by looking at the aggregate of underlying de novo peptide IDs. Here I'm showing a per position confidence in each amino acid of the CDR3. We can identify specific amino acids that have the lowest support and can potentially mutate them to obtain additional binders. In stage 2, we assemble full-length sequences. Now, rabbit antibodies pose a unique challenge for de novo sequencing. We're well acquainted with this challenge because we've observed hundreds of thousands of rabbit antibodies from B-cell sequencing. Framework 3 is unusually long and unmutated in rabbits, and in order to link together a CDR2 and a CDR3 accurately in an assembly, you need either unique mutations or long peptides to span the region. A key advantage of developing our own de novo sequencing tool instead of using something off the shelf is that it was developed and optimized to de novo sequence long peptides. Associating CDR2 and CDR3 is a problem akin to phasing in genome assembly. To illustrate this, imagine our sample contains 2 proteins, each 3 amino acids long, and we digest that protein into short, single amino acid peptides. In de novo protein sequencing, our goal is to reverse engineer the starting sequences from the set of peptides. If the peptides are short, we're missing a lot of information about where these peptides are located in the true protein. Without that information, we can assemble them incorrectly, and we have several different ways that we can get the assembly very wrong. With Griffin, we use longer peptides, and by using longer peptides, we can get pairs of amino acids that help us associate which peptides go where in the protein and arrive at the correct assembly. A second challenge in rabbit is the use of gene conversion to generate diversity. Most animals use somatic hypermutation to introduce amino acid changes to affinity mature, but in rabbits, whole parts of upstream pseudogenes are inserted into functional variable regions. We use a fully de novo sequencing approach to antibody sequencing, so we do not run into the challenge of mapping to and selecting peptides for a germline gene. Approaches that use a reference database will have a very difficult time sequencing rabbit antibodies. Here I'm showing an example assembly from our collaboration with Emory University. The depth of peptide sequences is shown here in all the rows in the alignment, and you can see we generate lots of peptides with lots of overlap. In a typical assembly, we're incorporating over 2,000 distinct peptides. The peptides are then assembled into a full-length sequence that's shown here in the MAB line, and I've noted the CDR1, CDR2, and CDR3, and you can see just how big that framework 3 region is between CDR2 and CDR3. I've also shown the alignment to the reference, which was not used in the assembly, but it shows where the mutations occur, primarily in the CDRs. We expressed the top antibodies and sent them to Dr. Kukar for testing. The assays he cared most about were ELISA and Western blot. So on the left here, he's comparing the specificity of MABs 1 through 8, the top 8 antibodies, to the original polyclonal. And you can see that the antibodies that are generated by Griffin are very specific to granulin 2 and also show reactivity to the intact progranulin domain at the bottom. On the right, I'm showing a Western blot assay that shows that one of our antibodies is able to detect both progranulin and granulin 2 domain in 2 different cell lysates, but not in the knockouts. So to summarize, Griffin is our new technology for polyclonal antibody sequencing. It's extendable to any species that has a backbone and a jaw, therefore an adaptive immune response. There are no B cells required, so we start from just plasma or serum, and what you get out of the process are validated antibodies that recapitulate the behavior of your polyclonal. At Abterra Bio, antibody sequencing and discovery is our bread and butter, and we'd love to talk to you more.